Today, I'm going to be your virtual lab instructor for a bacterial growth experiment. We're going to do this experiment in 3 simple phases. First, we're going to make our nutrient agar plates. Second, we're going to collect some samples. And third, we're going to plate those samples. So let's get to the lab. Alright, now that we're in the lab, first things first, we have to get our safety equipment on. That means gloves. Now that we have our gloves, we're going to need our lab coat. Lab coat done all the way up. Last thing is, that's right, eye protection. Okay, looks like we've got everything, so now let's get our reagents. First thing we need is LB powder. The second reagent we need is agar. And lastly, we're going to need some distilled water. Now that we have all of our reagents, let's head over to the weigh station. Okay, we're good to go. We've already filled up our container with 200 milliliters of distilled water. Then we're going to carefully take our reagent powders and pour them into the container. And now, for the most crucial step, we're going to take this container and we're going to shake. Shake, 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 shake. Once you're done shaking the first time, fill up the rest of the bottle with another 200 milliliters of distilled water. And then you're going to shake again. Shake, 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 shake. Shake it off, shake it off. What a workout. Once you're done shaking your solution extremely well, the next step is to have it sterilized in an autoclave. But maybe you should let your teacher or whoever's in charge of the lab handle this step because it requires a trained professional. So once your lunch break is over and the autoclave process is done, the agar is going to come out extremely hot, so we have to let it sit for a little bit to cool. Now while your agar is still hot, but cool enough just for you to handle, it's time to pour some plates. You're going to want to pour just enough to cover the surface of the plate, which is approximately 10 milliliters, but you can eyeball it. About everything for pouring our plates. Now you're going to let them sit and cool at room temperature, and if you're not going to use them right away, it's best to store them in the fridge at 4 degrees Celsius. You can pour as many plates as your experiment calls for. Now for sample collection, you're going to take your swab, and whatever surface you go to, make sure that you move the swab around completely to maximize any pickup of any critters you're going to get. Now once you've collected your samples, it's time to plate them. So take your swab, carefully lift the lid of the plate just enough so you can get your swab in there, and gently cover the surface of the agar. And that's it. You can do this same process for a whole variation of treatments. So once your samples have incubated for 2 days, be sure to check your results. And that's just about it for the rest of our experiment. Remember, be safe, have fun.